Gene electrotransfer is a physical method used to deliver genes into the cells by application of short and intense electric pulses. In successful gene electrotransfer, several steps are involved. Formation of a complex between DNA and cell membrane. Translocation of DNA across the permeabilized membrane. Transfer of DNA from cytoplasm into the nucleus and gene expression. For gene electrotransfer, different electric pulse protocols are used in order to achieve maximum gene transfection. One of them is changing the electric field orientation during the pulse delivery. In this video, we demonstrate the difference in gene electrotransfer efficiency when all pulses are delivered in the same direction, and when pulses are delivered by changing electric field orientation. Experiments were performed on Chinese hamster ovary cells, CHOK1. For the experiment, follow these steps. Grow cells in a culture flask in the incubator for 24 hours. On the day of the experiment, prepare cell suspension by trypsinization with 0.25% trypsin EDTA solution and count the cells. Centrifuge cells for 5 minutes at 1000 rotations per minute at 4 degrees Celsius and resuspend cell pellet in isoosmolar sodium phosphate buffer to a cell density of 5 million cells per milliliter and move the sample to the appropriate dish. Cells are exposed to electric field in pipette tip with integrated electrodes connected to a high-voltage prototype generator. These electrodes allow application of a relatively homogeneous electric field in different directions. The tip and the generator